Next up is a subject that's very dear to my heart anyway, is grass grub, seeing as I stopped using neonicotinoids about 5 years ago, and we've had a jolly good rogering this year with grass grub, so we've tried a few different things, but this is what I'm looking forward to. Thank you.
Thank you, Simon, and thank you for the opportunity to talk to you all today, especially for Alison's wonderful introduction, and I really endorse the asking of questions about biologicals. So it's great today to be talking to you about a new biological option for grass grub control, and I'll be co-presenting with Richard Chynoweth here from FAR, and we're talking on behalf of a number of co-authors as well from both Agresearch and also Midlands, who are another partner in this project. So what's this new biological active that I'm talking about? It's a species of Serratia proteomaculans, that occurs naturally here in New Zealand soils. And the strain that we're using is currently given the name of AGR 96X. So that's just the strain name. We don't actually have a trade name for the product yet. It has strong activity against grass grub and also against manuka beetle, which isn't much of a problem on the Canterbury Plains, but is a problem in parts of New Zealand. The way it works is the grub has to eat the bacteria for it to be infected and to show disease. And I'll see if does the laser pointer work? There we go, that blackened grub there, that's the sign of proteomaculans infection compared with the healthy looking grubs on either side. And the grub will stop eating very quickly, and then it takes, on average, about a week to actually die. But it stops eating and stops damage quite quickly. The way we're using it is a granule that's incorporated into soil, either direct drilled through a commercial product, either with seed or into established pasture. In some situations, you may be looking at a broadcast followed by a sort of a light soil incorporation. The key point is to get the granules in where the grubs are feeding. So it needs to be underground. And we are working towards a CVM registration. So I'm just going to give you a quick overview of 3 trials. Those are just a subset of quite a number of trials we've been doing. This one is a clover seed crop that was done back in 2019 through to 2020. And you can see here what we're showing is the yield of the different treatments. And consistently you'll see the red treatments are standard registered insecticides. The green here are a couple of different rates of 96X, and you can see the yields across all of these are very similar. And the untreated control has a much lower yield of clover seed at the end of the trial. So pretty pleased with that result. And here, this is a wheat seed crop. Again, the product was drilled at seed. And here we're looking at mortality. Richard will be talking more about yield. But you can see here the grub mortality in the treatments with the 96X are higher than the untreated control, and equivalent to the insecticides. The blue bar over here, this is actually your old Serratia entomophila, the same strain that you find in BioShield, which we put into this trial for a comparison. So this is the mortality you're seeing at 6 weeks after sowing, when we dug up grubs and looked at that. And this is a recent trial from the current season in established pasture, and this trial was done in Canterbury, as were the other 2 trials. And again, we're looking at grub mortality, and you can see the grub mortality with 2 rates of 96X are higher than the untreated control and equivalent with the insecticide. And this was drilled into an established pasture, so we're measuring it 6 weeks after drilling, 6 weeks after treatment. So what are we doing next? We've had multiple trials this season, which I don't have time to talk about. We've had some very positive feedback from the commercial contractors who are applying the product as it is now. There's a wheat trial that will be harvested next year, and we've currently got ongoing work around manufacturing, particularly around shelf life and storage, to try and get that up to a suitable standard for submitting the dossiers to ACVN for formal registration. And hopefully, if that goes well, we'll be looking to establish demonstration sites to trial the formally registered product. And I will pass over now to Richard to talk to you a bit more about this.
Right. Thank you, Sarah. And yeah, I'm going to talk to you about the trial. Some of you will have seen these results at the conference, FAR conference a couple of weeks ago. But this is the results that we put it through at Chertsey last year. You might have seen it there at the field day. So we've compared it against a number of different products, Poncho being the neonicotinoid, Suscon Green being an organophosphate insecticide, and then in combination with a few other things. So this is a BioShield formula. This is what we're talking about. The BioShield formulation is here on its own. It's a slightly different strain, I believe, in the trial that we had last year. And there's another treatment at the bottom here that might be of interest as well. It's Poncho plus a different product. So we established it last year on the 13th of May, which is quite late, but after lockdown. We weren't allowed to drill anything during the COVID lockdown. We drilled it into a site there at Chertsey, direct drilled into 300 grass grub larvae per square metre, targeting 150 plants a square metre a week. So effectively two grass grub larvae per seed of barley. So it's asking quite a big job to be done. So there's wheat is also, so right next door over here was wheat, and this is the barley. So you can see this is the 11th of August, 3 months after sowing, and the untreated is almost disappeared completely. The green treatments next door here, these both a Poncho treatment, seed treatment, and this one here's the Poncho followed by the Grub Zero treatment. So the Poncho gave us a lot of upfront protection. This plot right next door is the AGR96X scenario. So you can see a little bit of, there's definitely been some plant loss, and we do see that quite a bit. Actually quite comparable to Suscon Green. So up front we expect a little bit of damage, and you probably expect that when the grub have to eat the product to actually have an effect. So I think there's a few things around delivery that we could work on, but once you start getting the effect, then the grass grub definitely stop feeding, and the plants that are there, like Suscon Green, recover quite quickly, and you'll see in the yield data as we go through. So, not surprisingly, untreated's got absolutely hammered. We actually still managed to get 3 tonnes of barley out of that plot, which is surprising. And wheat actually has longer to compensate, so we produce a lot more tillers and wheat over time, so it did a wee bit better. The seed treatment and Suscon Green, or the true insecticide treatments, produced us about 10 tonnes of wheat, which is still quite respectable, and around about 8 tonne of barley. The biologicals, so the top one is the AGR96X alone. Statistically, these are the same as the treatments above, which, given the amount of pressure they're under, is a really positive result. The combination, this is a combination of the two, slightly lower yielding, but still statistically similar to the 96X. And that's a combination treatment that we've seen time in, time out, that they actually perform reasonably well like that. And this product here, so really effectively a Poncho treatment, I believe when we applied this product, the grass grub had probably stopped feeding. So overall, my experience is that we see a little bit of damage up front, just very similar, I'd put it very similar to a Suscon Green type effect, where up front you can expect a little bit of damage, and we might have to look at a little bit of how we deliver the product, whether we can put it slightly deeper than seed, so the roots grow into it, and bits and pieces like that in the future. Also, we'll probably look at using it a bit earlier in the season than May. So, when things are a wee bit warmer, try and get it to cycle a bit quicker in the soil. I think we'll take questions.
Is the carryover, will it carry on into the next drop once it's in the soil?
The AGR96X, that is.
Yes.
In an arable crop, no, because the cultivation process as you change crops means you'll lose that. In pasture, possibly. We've only got one trial where we've been able to sample for a second year, and it's suggestive. We will be looking at the pasture trials we did this year. We're going to do follow-up sampling and see if it does persist. So,
what about direct drilling situations in arable? You say cultivation destroys it, but if you're direct drilling, you're not cultivating, so...
We haven't attempted to look at that in an arable crop, but we wouldn't, I would be surprised. I haven't seen any evidence that, for example, Serratia entomophila, which is another naturally occurring organism, it tends to not persist between crops in the arable system. So I would probably not expect it to happen, but I won't rule it out if we try it.
We'll be looking at it at the site we've got at Chynoweth Farm in Lincoln this year. So the trial's been established there, and we'll be looking there with a hope that we can direct drill some probably nipping grass through that, and see if it carries over from one year to the next.
In the earlier slides, there were two, what I saw as anomalies there. One was that the biocontrol worked better compared to the conventional ones in the wheat than it did in the clover. But so there was more control in the wheat than there was compared than there was in the clover. And then the other one was that I think it was in the wheat. 30 kilograms to the hectare was not as good a result as 20 kilograms. Is there an explanation for that?
I don't think we've had 20 kilos on its own of AGR96X in the wheat. So that was a combination treatment with the other Serratia. I don't recall seeing the clover treatments, but what I could imagine there is that it's actually susceptible to UV light. So if you don't get good soil coverage with in the clover where you're planting it nice and shallow, then you could see a reduction. And whereas in the wheat we're planting that down 5 centimetres, I don't expect there's any loss through that sort of mechanism in that time frame.
So is it a fungus? No.
No, it's a bacteria.
A bacteria, right?
Yeah,
Okay.
it's a bacteria.
Yeah. So is there anything on the agchem that we use conventionally that would blow it out?
We don't have any evidence for issues with that, and certainly, thank you. Sorry, in this year's wheat trial, we've included some combination treatments where we've included 96X and other currently registered insecticides, and there's no evidence of any detrimental effect. So no, I don't think we would expect it, particularly because the granule itself is separate from the insecticide treatments. You drill it alongside with the seed, but it's not incorporated with the seed or incorporated with any other treatment. So.
And just to add to that, we did do some work, I think it was 2013, where we actually seed coated the BioShield formulation of Serratia on top of Poncho with a barrier between them, and that acted perfectly fine in the field and did the same sort of job as we expected there. The problem with that one is it might take 3 to 4 weeks before you start to see any death, and cropping that's just too long if you're asking the product to work at sowing. If you applied it pre-sowing, then you might get some added benefits of being able to get death before you plant your crop.
So the AR96, it couldn't be applied as a foliar to be washed in a rain with being so UV sensitive by the sounds of it.
Yes, it is UV sensitive, and it is actually a soil dwelling bacterium, so it's not one that's capable of persisting on foliage. Yeah, you'd lose too much. So.
Could it be done as a liquid in FAR?
We haven't attempted that, but of course originally the old Serratia entomophila was a liquid drilled in furrow. We're going with the granule in part because the granule ingredients actually are both attractive and tasty to the insect, and you need them to eat to get the bacteria, but it also actually acts as a food source for the bacterium to allow them to grow and help them sort of stay alive and persist in the crop. So that works better, we think, than using the liquid. Plus the liquid potentially requires different machinery, whereas this granule can go through conventional seeders.
You say the grub's got to eat it, then is it taken up by the plant and then...
No, it's not. No, they have to actually eat the bacterium, either ingesting incidentally in soil particles or through actually eating the little granule that's been drilled. So it doesn't go into the plant at all. We've actually tested that as part of the sort of phytotoxicity and so on in preparation for registration. It's not something that's taken up by the plant.
I'm trying to get my, so I don't have a huge amount of experience with grass scrub, sorry Nigel. So do grass scrub eat soil as well when the...
As they move, they do.
As they move, they do, yes. The grubs are root feeders, but as anyone who's looked at them, you can actually see they sort of ingest soil incidentally along the way and as they're burrowing through. So yeah, they do pick up soil, and that's naturally how Serratia entomophila causes amber disease in wild grass grub populations. You do get natural outbreaks of that, and there they are just ingesting it accidentally with through the soil or through contact with dead infected grubs that of course shed more bacteria. So yeah, that is the natural disease cycle. Sorry.
Is your application rate, you mentioned 30 kilograms a hectare. That's quite a high application rate for something you're trying to put in with a sowing rate. Like I've spent a lot of time putting a plethora of different biological things through a small seed box, basically designed to put in green insecticide. Just thinking in terms of the practicalities and something that's going to be a barrier for farmers to do this, if you're stopping every couple of 3 hectares to fill up your insecticide box, when your wheat goes 10, 15 hectares at the stop, is there any chance of lowering that rate, or has that become the issue with the grub running into the soils you need? I guess what I'm trying to ask is, can you have a higher rate, so you need a lower application rate to make it practical?
Yeah, unfortunately, from what we can see, the lowest 20 kilos may be effective in some cropping situations. Well, not so much cropping, but in pasture, 20 kilos might be enough. For most cropping situations, you've got a lower damage threshold, so you can tolerate fewer grubs per square metre. And that means you've got to have that higher application rate, because you've got to get enough granules. If you imagine the grubs as little targets, you've got to get a granule near each grub. And if you push it down too low, you're not going to have sufficient interception to prevent damage to your crop. And that's particularly true if you think of, say, something like carrot seed crops that have a lower sowing rate and have a very low threshold for grass scrub. They can only tolerate, it's 50 grubs per square metre is a damaging threshold for some of our valuable seed crops. And I would say we have tested these, at least in pasture, with very large plots, looking at 600 square metre, and also a single trial with plots of larger than a hectare, and the commercial applicators were able to use it. They were able to use 30 kilos per hectare. So.
Just to add to that, we may have to rethink a wee bit with grass scrub in the future and actually plant more seeds and actually be prepared to lose a few. The other option would be to look at doing that. So at the moment, like our trials, we're still targeting the standard sowing rate, expecting that we're going to reduce plant numbers, but why not plant a few more and actually expect that we might lose a few along the way.
2 questions. One is the temperature at which the bacteria is active, and also is there any opportunity to add supplementary nutrition to build up the population?
Supplementary nutrition to build up the bacteria population? Yeah. The granules actually do that themselves. The bacteria, once the granules are put into the soil and pick up a little bit of moisture, the bacteria start growing. So they actually multiply very quickly on that granule. The other thing that happens to keep the bacteria going is that once they infect the grubs, they will, we do think there are some disease cycles, so that a dead grub will recycle and infect others. And I'm sorry, remind me what your first question was?
Temperature of activity.
The temperature, okay. So our main barrier is not so much temperature in terms of the activity of the bacterium. It does obviously not want to be very hot, as Alison mentioned. The bigger problem is the activity of the grubs if you apply it too late, because as it gets colder, they're feeding less and they themselves are less active, so they're less likely to encounter the bacterium. The bacterium will still be active if they meet it, but it's more about the temperature in relation to grub activity.
One more question.
One more question.
I was just curious to know, you've partly answered my question now, but when you put the bacteria in the soil, I know you've just said that you've got food in the greenhouse for it, but would plant exudates feed that bacteria?
We haven't looked at that. The bacteria does occur naturally in the soil anyway, so it appears to be able to survive naturally in soils. The difference with Proteum maculans is it seems to have its natural background level is comparatively low, and you don't see natural outbreaks occurring in grass grub. Whereas Serratia entomophila, you do see naturally occurring outbreaks without human intervention. So as far as we can tell, it's capable of surviving on the soil on its own anyway. It's just unfortunately doesn't occur naturally at a rate that's useful for our purposes.
What I would find interesting research would be if you had a carryover crop between, like the wheat, what you're saying with cultivation, you're destroying it. What would happen if you had a carryover crop so you don't have to keep re-inoculating the soil?
Unfortunately, this bacterium isn't one that lives on plants. So,
well, I mean,
yeah.
if it's getting fed from the plants or...
Yeah, there's no evidence that it does that, I'm afraid. That's not its host is grass grub, and yeah.
So what does it get fed on?
I don't know, natural nutrients, I guess. Yeah, it's not associated with plants. When we sample for it, we literally dig up soil. We don't look at plant roots. That's us for now.